Today we're going to be looking at our brief intro to cells. Now, I'm sure you've memorized all your cell parts and you still know them by heart from freshman year biology, but we're going to review a little bit more in detail some of our organelles and why cells are so important. First, let's take a look at our cell theory. So cells are the fundamental units of life. So all living things are made of cells, and all cells come from other cells. And these three main parts took scientists a lot of years to sort of agree on these three parts of the cell theory. And different scientists like Theodore Schwann, who we call Schwann cells after, came up with different parts of this theory, but this is the essential and fundamental parts of it that we know now. Before we get into surface area to volume ratio like we were talking about today, let's talk a little bit about how we can see these cells. Now, there's different types of microscopy that scientists use to visualize cells. We are mostly going to be working in this class with light microscopy using our light microscopes, but there's tons of other different types of microscopes you can use as well, and some of our prettiest pictures we're going to get frequently from electron microscopes, such as our scanning electron microscope, which is just going to sort of look at the surface of a cell. Now, some cells are really, really tiny, and if we look at the scale of cells, certain bacterial cells are only going to be a few micrometers long. And so our scanning electron microscope is going to be really useful to see and visualize parts of these cells. And of course, most of our subcellular structures, including our organelles, are too small to be seen by a regular old light microscope. So that's why we have to use either a transmission electron microscope or a scanning electron. And these are abbreviated by SEM and TEM. You can check out the types of microscopes video for more detail on that too. So, with recent advances in microscopy, we have new techniques for different types of microscopy. And one of those is cell fractionation, and I'll put that on the screen. And that's where we're taking apart the cells to separate major organelles from one another. And so we're going to use a centrifuge. We'll fraction it off into parts, and then we use this fractionation to determine the functions of the organelles. Okay, so today or some other day in class, we talked about how cell size is really important. Why are we not just one big cell? In fact, it has to deal with the surface area to volume ratio. Cell form relates to function. So our cells are so many different shapes because of the fact that we need to get molecules across the membranes of these cells and into the cells efficiently. If the cell got too big, it just wouldn't be efficient, and we wouldn't be able to get the molecules in or out that we needed in enough time. When we look at our surface area to volume ratio, we're going to see that the cells that have more surface area just have more points across them where molecules can diffuse. This is a bad-looking neuron with some dendrites that I drew. But cells that have more efficiently evolved to certain shapes can do their jobs a lot more efficiently with a higher surface area to volume ratio. So a cell with a surface area to volume ratio of 12:1 is much better than a cell with a surface area to volume ratio of 1:1. That's not as good. And we'll see in our lab where we use the agar cubes how it's going to take a lot more time for certain molecules to diffuse all the way to the center in a cell that has, or a mock cell that has, a smaller surface area to volume ratio. Okay. So, moving on to our two main types of cells. Now, all of life is separated into our three domains of life, our archaea and our bacteria, and our eukarya. Archaea and bacteria belong to what we call our prokaryotic group, and our eukarya, of course, belong to our eukaryotic group. Now, you guys probably remember the few main differences between prokaryotes and eukaryotes. Prokaryotic cells do not have a nucleus. Instead, their DNA is contained sort of in a clump in the cell called a nucleoid. Eukaryotic cells do have a membrane-bound nucleus. They also have membrane-bound organelles. There are really no true membrane-bound organelles in our prokaryotic cells. We'll see later on that certain membranes can appear in some prokaryotic cells inside the cell, but we'll talk about that a little bit later. Prokaryotic cells do have ribosomes, they do have a cell membrane, and some of them actually have evolved more complex structures like a flagellum, excuse me. And eukaryotic cells are much more complex. They have all these organelles, they're compartmentalized, and they have evolved to have different parts of the cell do different jobs. So our protists, our fungi, our animals and plants all consist of eukaryotic cells. We are eukaryotes, you like you and me. Prokaryotics, pro means more primitive, and of course these are the cells to first evolve. We as scientists believe that eukaryotic cells came from prokaryotic cells through the theory of endosymbiosis, where one day one prokaryotic cell sort of just swallowed or took in another or had some sort of symbiotic relationship with another smaller prokaryote. And we have evidence for this through the DNA that appears in organelles such as mitochondria. And we actually theorized this before it was confirmed, but there is such a thing as mitochondrial DNA, and we can trace that through a mother's line and actually see the exact same DNA appear from mother to child over generations and generations. It's very interesting to follow that, but we'll talk more about that when we get to our evolution. So again, all cells have a plasma membrane. They have our sort of semi-fluid substance called the cytosol. They have chromosomes or DNA, they carry genes, and they have ribosomes. Now let's get to the structures that just, these are the simple things that prokaryotic cells have. So again, like I said, their DNA is contained in a nucleoid. Apologize for my bad handwriting here. The cytoplasm is the rest of the material in the cell besides the DNA. So the cytoplasm will include the cytosol, which is like our water, ions, different organic molecules that are doing their jobs. Ribosomes are what are going to can't talk tonight, guys, carry out protein synthesis. We also have cell walls, and these are particular structures that are pretty useful for our bacteria. We know that we identify a lot of bacteria through what's going on in their cell wall, such as like gram-positive and gram-negative bacteria. They also have some internal folded membranes that can carry out certain cellular processes. And you can read a little bit more of these in your text. But these are interesting because these evolved a little bit later after our original prokaryotic cells first appeared. Flagella, of course, have motor proteins in them to help them move. And we see flagella in both prokaryotic and eukaryotic cells, such as sperm, but bacteria also have these as well. And we start to see a cytoskeleton, so more of an internal structure in parts of our later to be evolved prokaryotic cells. All right, so let's talk about some of these membrane membrane-bound organelles that are going to appear in our eukaryotic cells. We have our endoplasmic reticulum, which we can abbreviate through ER, RER, SER. Our SER, smooth endoplasmic reticulum, is going to do things like synthesize lipids, metabolize carbohydrates, sometimes detoxification, and store calcium ions. So that's very important as well. Our rough endoplasmic reticulum has bound ribosomes, so certain types of ribosomes will attach here, and there will, of course, ribosomes are the things that are going to help us make our proteins. And so proteins that belong on the rough ER will be made by those attached ribosomes. So those ribosomes are going to create glycoproteins, and then they're going to distribute transport vesicles, and generally sort of a membrane factory for the cell. A lot of the proteins that are going to end up on the plasma membrane will come from the rough endoplasmic reticulum, so that's very important to remember. Our Golgi apparatus is going to consist of a lot of sacs we call cisternae. And this is going to modify products produced by the endoplasmic reticulum and manufacture other macromolecules and just sort and package materials. We call this like the post office of the cell. Lysosomes are digestive compartments. So a lysosome is like a membranous sac of enzymes that can digest larger macromolecules. There are certain enzymes that work best in the acidic environment of the lysosome, so that's why we have them. The inner material of the lysosome is a little bit more has higher acidity than the rest of the cell in general, but it can take care of a lot of different macromolecules, which is great. All right, so let's talk about some of our energy producing or energy involved organelles. Okay. So this is a mitochondria in here, plural mitochondria. We have these in nearly all eukaryotic cells. They have a smooth outer membrane and an inner membrane folded into what we call cristae. So instead of little zigzags like we draw when we're drawing an ugly mitochondria, we'll call these cristae now. And the inner membrane has two compartments. We have an inner membrane space and then a mitochondrial matrix. And a lot of the steps of cellular respiration, we have to recognize where these steps are going to occur. But some of them are catalyzed in the mitochondrial matrix. And our cristae are going to use or give us a large surface area so that enzymes can synthesize ATP, produce that energy that we need, because our energy is cellular ATP. All right. Then we have what's called our plastids. So this is a group of organelles like our chloroplasts, but we also have chromoplasts and leucoplasts. And so our chromoplasts are going to exist. They'll give us a different color pigment, sometimes like a yellowy pigment. Our leucoplasts are more for like starch storage. But these are also involved in things such as our energy processes. Of course, our chloroplasts are really key for photosynthesis, but these will show up in other plant species as well. All right, time to talk about some more digestion. So peroxisomes are going to break down our toxic products that our cells produce, such as hydrogen peroxide, H2O2. So our peroxisomes are specialized metabolic compartments. They're bound by a single membrane, and generally they're going to break down toxic things, mostly hydrogen peroxide, and convert it to water, because we can take this apart and make it harmless if we digest it in the right way. They have lots of reactions, but it's still not quite sure exactly what peroxisomes do with other organelles. So there's still some questions surrounding the roles of peroxisomes. Okay, these other two, these are going to show up in young plants. Vacuoles, of course, are for storage, and also some structure. We have one large vacuole in plant cells, but mostly we'll use it for storage in our eukaryotic animal cells as well. All right, time to talk about structure of the cell. I was getting ahead of myself for a while. So we have microfilaments, intermediate filaments, microtubules, and cilia and flagella. These, of course, are created from specialized proteins that are going to give us motor movement of the cell. We know that cilia and flagella are going to provide important movement because when we remove them from the cell, they can no longer move. All right, our main roles of our microtubules are going to be maintaining cell shape, obviously, because it's for structure, help us with motility. Sometimes they'll appear in cilia and flagella, and they can help with chromosome movements and cell division, and of course, movements of organelles if we need it. Our intermediate filaments are going to have a lot of roles. Some of them are maintenance of cell shape, helping anchor the nucleus, and then formation of the nuclear lamina, which is that surrounding protein portion of our nuclear envelope. And finally, our microfilaments have lots and lots of roles. They'll help us with muscle contraction, they'll help us again with motility, like in a pseudopod, help us with cell division, help us with changes in cell shape, and just general maintenance of our cell shape. Okay, finally, let's talk about cell walls. Now, some prokaryotic cells have cell walls. Plant cells do, animal cells do not, of course. It's an extracellular structure that is going to help us distinguish from plant and animal cells. So some protists have these, some fungi have these, but again, animal cells, our cells, we do not. It's going to help protect the plant cell, help maintain the shape, and also prevents excessive uptake of water. So plant cell walls are mostly made of cellulose fibers. Cellulose, which is hard for us humans to digest. So plant cell walls have lots of layers. There's like a primary and a middle and a secondary, but we will move forward and talk finally about the extracellular structure, the extracellular matrix of animal cells. So animal cells don't really have cell walls, but they do have our extracellular matrix, and it's made up of like glycoproteins like collagen and fibronectin, and it's going to bind to receptor proteins in the plasma membrane, and this is going to help us have support, adhesion, movement, and regulation in the cell. So when we start talking about cell junctions is when we're going to start talking about cell communication, which will be hopefully one of our next few videos. But there's lots of types of cell junctions that we will get into, and hopefully you guys are excited to hear about those. All right, so again, please go back and review these parts of the cells. Make sure you do have notes for every single organelle we have covered. We're going to be talking about them in class. Thank you, guys.